Welcome children. Welcome to another chemistry class. Today we are discussing the chapter Biomolecules. One of the simple chapter where the main discussion is about the carbohydrates, then about the proteins, DNA, RNA, etc. That is the only minimal point we need to concentrate on the chapter. Okay, let's have a look at the important questions. First one, a fundamental question which is the biology students definitely know that central dogma of molecular genetics states that the flow of genetic information, all of you know DNA to RNA to proteins, all of you know that it is DNA to RNA to proteins. First choice. Yes. Central dogma states that pattern of information that occurs most frequently in our cell from existing DNA to make a new DNA replication. From DNA to make a new RNA, transcription. From RNA to make a new proteins, translation. All of you know that. Next. The correct corresponding order of names of 4 aldoses with the given configuration. It is erythrose and thriose are given. Erythrose and thriose are given. You see, when the two OH groups on the chiral center on the same side is called erythrose, opposite side is called thriose. So it is basically diastereomers. It is basically diastereomers, and the answer is second choice, D-erythrose. You see, both OH on the same side. That is erythrose. And what is D? On this carbon, functional group to right. That is called D-erythrose. So what is L-erythrose? If this is D-erythrose, this is L-erythrose. Right? The functional group on this carbon should be on to left hand side. That is L configuration. Erythrose means all the OH group onto the same side. So this is D-erythrose. That will be L-erythrose. They are enantiomers. Now OH on the opposite side. That is called thriose. And now once again on this carbon coming to right hand side. That is D configuration. On the same carbon it is coming to the left hand side. That is L configuration. So it is D-erythrose, D-thriose, L-erythrose, L-thriose. That is the answer, second choice. Right? Next one. Not correct. Which is not correct? First one, correct statement, all of you know. Second one, correct statement, all of you know. Third one, correct statement. The fourth one is the incorrect statement. This is an incorrect statement. This is correct, this is correct, this is correct. What is incorrect in the fourth one? Denaturation make the proteins more active. Wrong. Denaturation spoils the enzymatic activity. During denaturation, the secondary, tertiary, quaternary structures are lost and is reverting to primary structure. Basically, the well-defined geometry due to tertiary structure is the fundamental principle behind the what? The enzymatic action of proteins. So, activity of proteins is lost. The native character of protein is lost. Right? So, the answer is option fourth choice. While all others are known to you. Next one. Which can form sweeter ion? Glycine. Glycine is an amino acid that can form sweeter ion at isoelectric point. Glycine is the first amino acid and that can form sweeter ion. And this is the sweeter ion formation of glycine. This is the first amino acid glycine, and this salt and this acid group and amino group can exist in the sweeter ion form, dipolar internal salt. What is the pH concerned known as? Isoelectric point. Next question. Difference between amylase, amylopectin. Amylopectin 1,4 linkage, 1,6 beta. This is wrong. There is no beta linkage. Amylose 1,4 linkage, 1,6 linkage. This is wrong. Amylose is a straight chain polymer. Amylopectin 1,4 linkage, 1,6 linkage. This is the correct statement. Amylopectin is a branched molecule. Branched polymer. Highly branched polymer. And what is the kind of linkage? Reference CRT. In the straight chain, it is going for 1,4 glycosidic linkage, alpha glycosidic linkage. And during the branching, it is making 1,6 alpha glycosidic linkage. Both are alpha glycosidic linkage, and the straight chain go for 1,4 linkage, and the branching go for 1,6 linkage. Okay, that is the correct answer. Amylose, that is the wrong answer. That is also wrong. This is not correct. Galactosilla. Next one. Yes, this is what the linkage in amylose and amylopectin. Amylose will go for 1,4 glycosidic linkage, linear polymer. Amylopectin will go for 1,4 glycosidic linkage in the linear chain and 1,6 glycosidic linkage in the branching. Okay, next. Non-essential amino acid. That is another important point you need to understand. Essential and non-essential amino acids. There are 20 natural amino acids. Almost 10 is essential, 10 is non-essential. Essential one means you have to take it through the diet. Non-essential means it is produced in the body, all of you know. Right? The non-essential amino acid is leucine, alanine, lysine, valine. Alanine, it is alanine, second choice. These are the essential amino acids. Arginine, isoleucine, histidine, leucine, methionine, lysine, phenylalanine, tryptophan, threonine, valine. They are the essential amino acids. Which are all non-essential? Alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, proline, serine, tyrosine. They are all. So this is one of the most important area concerned to amino acids. Essential, non-essential amino acid. You must learn that. Okay. Next. Sucrose on hydrolysis gives. Sucrose will give you what? Alpha glucose and beta fructose. Alpha D glucose and beta D fructose. Alpha D glucose and beta D fructose. Second choice. Second choice. This is sucrose. Alpha D glucose and beta D. Oh my God. It's not beta D glucose. It is beta D fructose. Fructose. Next. Which is a basic amino acid. See, that is another classification you must know. Acidic amino acid, basic amino acid, neutral amino acid. It depends on the number of amino group and COOH group. If they are equal, it is neutral. If COOH number is greater, acidic. If NH2 group is greater, basic. All of you know. So you need to know which is basic amino acid, and the answer is lysine. Lysine is a basic amino acid, and this is the structure of lysine. There is one COOH group, while you have two NH2 group, it is basic. What about alanine? There is one acidic group and one basic group, neutral. What about tyrosine? There is one NH2 group, one COOH group, that is neutral. What about serine? There is one COOH group, one NH2 group, serine. So all others are neutral characters, while lysine contain two NH2 group, one NH2 group extra. Okay, next. General formula of carbohydrate. CxH2O times. That is how the name derived. Carbohydrates. CxH2O times. Most of the carbohydrates are able to written by this formula. But this is only traditional and conventional. Don't think that. Those who are read NCERT clearly, you might know that. There are many carbohydrates which does not fit into this formula, raminos. There are other molecules which fit into this formula and not carbohydrates. Acetic acid. Acetic acid formula fit into this general formula, but not carbohydrate. Raminose formula does not fit to this general formula, but that is a carbohydrate. So there are exemptions also. Next one. Two monosaccharide units in maltose. In maltose, what is the monosaccharide units? All of you know. Sucrose, maltose, lactose. We are learning. Sucrose, maltose, lactose you are learning. And this is maltose, alpha D glucose, 2 alpha D glucose. And it is option A. 2 molecules of alpha D glucopyranose constitute maltose by 1,4 linkage, and the functional group is free here, and it is a reducing sugar. All of you know. Next one. Glucose with the excess of phenylhydrazine forms. Glucose with excess of phenylhydrazine forms. Glucosazone. Ozazone is formed. Ozazone is formed. Glucose can react with not only glucose, fructose also can react with the 3 molecules of phenylhydrazine, giving the same product, ozazone. Whether glucose or fructose, you get the same product. What is the reaction? It reacts first with one molecule. It is condensing over here. You are getting this product. Again it is reacting. Again it is reacting. And it is interacting. And it is interacting over this region. It is interacting over this region. One of the hydrogen removed and one of the hydrogen removed, and it is converted to aniline and ammonia. And thereby you are getting CO group. And now it is again reacting over this CO group, condensing here. You are getting this one. This is called ozazone. Ozazone. And remember, try writing the reaction with the fructose also. With the glucose and fructose, you get the same ozazone. 3 molecules of phenylhydrazine will react and you get same ozazone. And the answer is option D. Next one. Alpha D glucose, beta D glucose, different configuration on C1 carbon, and they are called anomers. Alpha beta glucose are anomers. There is a change in configuration over C1 carbon. Anomers. Option B. Next. Number of chiral carbon atom in alpha glucose is. Number of chiral carbon atom in alpha glucose is. How many chiral carbon atoms are there? In the open chain, 4 chiral carbon atom. In the closed chain, 5 chiral carbon atom. And the answer is 5. 5. This is the glucose structure. And the chiral centers are 1, 2, 3, 4, 5. So if 4 and 5 given, you must select 5 because it is mostly existing in the cyclic pyranose structure. Okay, and there are 5 chiral centers. Next. 1 gram pure alpha glucose crystals are dissolved in water, and after attaining equilibrium, amount of alpha glucose in the solution is approximately, you might have heard about that phenomenon, mutarotation. Mutarotation, a spontaneous change in the optical rotation observed. That is called a mutarotation. And what is happening there? What is happening there? See, have a look at. A solution of alpha D, beta D glucose will form an equilibrium mixture of 1/3 alpha and 2/3 beta. 1/3 alpha and 2/3 beta. The rotation of alpha increasing from, the rotation of alpha decreasing from somewhat above 100 to 57 something, while that of beta increasing from 1920 to 50. So it will attain a equilibrium point. It will attain an equilibrium point, and which is so called mutarotation. Mutarotation. And that is about 36% alpha glucose is formed. 1/3 alpha and 2/3 beta. 1/3 alpha means some 33%. And the close figure is 36% approximately alpha glucose will be in the mixture. Now 36 percentage of 1 gram, that is 0.36 gram. So alpha will be 0.36 gram and the remaining will be 0.64 gram will be beta. That is the equilibrium existence in the solution, right? And you can see there is a natural change in the optical rotation. And the equilibrium value somewhat 50-52 degree rotation, it come and stabilize. Next. Lactose contain. Yes. What is there present in lactose? It is beta galactose and beta glucose. Option D, beta galactose and beta glucose. Lactose, milk sugar. We are learning in the NCERT 1,4 glycosidic linkage producing sugar. One is galactose, one is glucose. Next. Highly branched among the following. Amylose is linear. Amylopectin branched. Glycogen is also branched. Answer is option D. Glycogen, the so-called animal starch, it is highly branched. So amylopectin is branched and glycogen also branched. Okay, next. Amino acids show minimum solubility when the pH is close to. You see, amino acids we are separating at isoelectric point because at isoelectric point it exists as a zwitterion and it is more like a salt. See, amino acids are basically not giving the properties of amines or acids. They are showing the properties of salts. And that salt character mainly exhibited in the isoelectric point. A pH called the isoelectric point, and that is the point where we separate them in particular, and the solubility is minimum. It is separated as crystalline substances. Option B is the correct answer. Next one. The forces of attraction that stabilizes the tertiary structure of protein. Tertiary structure of protein. All are the cause of stability for the protein structure. Reference CRT. Next. A sample of DNA was completely hydrolyzed. The number of adenine bases obtained will be more or less equal to the number of. See, adenine pairs with thymine. So, number of adenine and thymine will be equal. Number of cytosine, guanine will be equal. So, adenine will be equal to thymine. Option A. Cytosine will be equal to guanine. Not found in nucleic acid molecule. Not found in nucleic acid molecule. There is adenine, there is guanine, there is cytosine, there is no thyroxine. This is not present in nucleic acid molecule. Nucleic acid molecule contain a sugar moiety, a nitrogenous base, purine, pyrimidine bases, and a phosphate group. And what is thyroxine? Thyroxine is a thyroid hormone. It's a hormone. Option D is the correct answer. Next. Pyrimidine derivative. Pyrimidine derivative. What are the pyrimidine derivative? Which of the following is a pyrimidine derivative? A and B. A and B. Uracil thymine, isn't it? Uracil thymine, isn't it? Pyrimidine, purine, pyrimidine bases, uracil, thymine, cytosine. They are the pyrimidine bases, nitrogenous bases. So, purine, pyrimidine bases, which are involved in RNA, DNA. In RNA, there is uracil. In RNA, there is uracil. Instead of thymine, there is uracil. While in DNA, adenine pairs with the thymine, cytosine pairs with the guanine. All of you know. Next. Which is optically inactive. Which is optically inactive. See, among the amino acids, there is one optically inactive. The first amino acid, glycine. Glycine is optically inactive. While all other amino acids are optically active. Answer is option B. Next. which contain a metal atom. Vitamin B12, cyanocobalamin. It's a complex of cobalt containing cobalt. Cyanocobalamin, that is vitamin B12, causing pernicious anemia. Next. Beriberi caused by. This is another area from the chapter. Even the latest NEET examination, there was question from that area regarding vitamins. A tabulated information given in the NCERT regarding vitamins. Learn that properly. And beriberi caused by, all of you know, thiamine, vitamin B1. Option B is the correct answer. Beriberi. Which of the following gives an optically inactive aldaric acid on oxidation with a dilute nitric acid? Option A you want to get. Option B is the answer. Option B is the answer. You will be getting, this group is converted to COOH and this group is also converted to COOH. And there is no changes in the other area. You are having like this only. And see, this molecule is now optically inactive. This molecule is now optically inactive because over this plane you can find a symmetry. Over that plane you can find a symmetry. These two parts of the molecules are same. And it is a meso compound. It's a meso compound and that is optically inactive. That will not happen over here because OH to right and OH to left. That will not happen here. OH to right, OH to left. That will not happen here. OH to left, OH to right. While here, both the OH groups are on right hand side. Therefore, upon oxidation, when the top CHO converted to COOH and bottom CH2OH converted to COOH, the molecule turn meso character. Okay, next one. Answer is option B. This is the correct answer. Next question. Pyranose ring consists a skeleton of. Pyranose ring consists a skeleton of. Pyranose is a 6-membered ring. There are 5 carbon atom and 1 oxygen atom. 5 carbon atom and 1 oxygen atom. Option A is the correct answer. 5 carbon and 1 oxygen. Option A. See, carbon 1, 2, 3, 4, 5 optically active carbon and 1 oxygen. Option A. Next one. Periodic acid splits glucose and fructose into formic acid and formaldehyde. Ratio of formic acid and formaldehyde from glucose and fructose is. That is the question. Periodic acid. Periodic acid means HiO4. HiO4 is called periodic acid, known as malapride reagent. Malapride reagent. It is basically 1,2-glycol breaker. 1,2-glycol breaker giving simultaneous oxidation. Not necessarily glycol, aldehyde keto groups also present at 1,2 position. It will break and give you oxidation product. So what is obtained in the first case? You will get 5 formic acid and 1 formaldehyde. How 5 formic acid? This is converted to formic acid. This is converted to formic acid. 1, 2, 3, 4, 5. You are getting 5 formic acid and this is a primary alcohol group will be converted to formaldehyde. Now what about with the fructose? With the fructose, this will be converted to formaldehyde and this will be converted to formaldehyde. You will get 2 formaldehyde while this will be converted to carbon dioxide. While this one will be converted to 3 formic acid. So with the glucose, you will get 5 formic acid and 1 formaldehyde. See the ticked 5 carbon converted to formic acid and primary alcohol group converted to formaldehyde. While with the fructose, the top and bottom carbon will be converted to formaldehyde and 3 ticked carbon will be converted to formic acid, and the CO group will be converted to carbon dioxide. And now what is the question? Ratio of formic acid and formaldehyde from glucose and fructose. In glucose it is 5:1, and in fructose it is 3:2. 5:1, 3:2. Option B is the correct answer. Right? Next one. Natural carbohydrates and amino acids are usually, natural carbohydrates are D configuration, while amino acids are L configuration, D and L respectively. Natural carbohydrates are D configuration and amino acids are L configuration. Remember, DL configuration are relative configuration based on which molecule? Acid. Which molecule? Based on glyceraldehyde. Based on glyceraldehyde molecule. Okay, right. (29) The number of chiral centers present in glucopyranose and fructofuranose are. Glucopyranose already we have seen. There are 5 carbon atoms. And fructo, how many it will be? 4 only. Have a look at the structure. This already we have seen. 1, 2, 3, 4, 5. What about fructofuranose? It is 1, 2, 3, 4. There are 5 and 4. Option B. Next question. A linear polymer of alpha glucose is amylose. Amylose only, because this is branched and this is also branched. Cellulose is not branched, but it is beta glucose. Cellulose is beta glucose. What is asked here? Alpha glucose. Alpha glucose only one, amylose, option A. While amylopectin is branched in nature. Cellulose is beta glucose. Right? And that is the end of questions from the concerned chapter. As I said, the most important area, structural details of carbohydrate, then amino acids, proteins, DNA, RNA, vitamins. There is an additional topic, hormones also. You can expect questions from that also. Okay, that's all from the concerned chapter, biomolecules. Okay, we'll meet in another class. Right, bye, see you, take care.